Welcome back to Dissect the Question. A group of researchers are working on the hypothesis that a mutation in the ryanodine receptor protein is a key factor in epilepsy. They're using cultures of human skeletal myocytes as a control preparation and comparing the effects of various ryanodine receptor agonists and antagonists on functions of myocytes and neurons. Which of the following best describes the response that is expected from administration of a ryanodine receptor agonist to a culture of skeletal myocytes? The key to the question is ryanodine. We can see in the clinical vignette there's a mutation in the ryanodine receptor that's being studied. And here in the lead-in, the question itself refers to ryanodine receptor agonist. An agonist is something that stimulates the ryanodine receptor. It binds to it and stimulates it. Where an antagonist is the opposite. It binds to it, but it inhibits it. So let's see. Where do we find the ryanodine receptor? So if you remember, our sarcomere runs from Z line to Z line. That's the contractile element, or the contractile unit of skeletal muscle. And we have thin actin filaments attached to the Z lines with a thick myosin filament in between. And it's the cross-bridging that occurs between the actin and myosin filaments that pulls the Z lines closer together to produce active tension, which is the force. Tension is a force for purposeful movement. And we'll recall that purposeful movement is when we have contraction and relaxation both, so that we can move kind of cyclically. But if we have contraction and no relaxation, like in a spasm, we would call that non-purposeful movement, because we can't use that movement to move a load or to do any useful work. So where does the ryanodine receptor come in? Well, to get a contraction, we first need a depolarization, an action potential, and the action potential comes from a motor neuron that releases acetylcholine onto acetylcholine receptors, opening a sodium-potassium non-selective cation channel that allows sodium to rush in. And then when sodium rushes in at the Z line, we call that depolarization. And that depolarization releases calcium from the ryanodine receptor calcium release channel. So here we have ryanodine receptors releasing calcium to allow that cross-bridging we just talked about to occur. That's what's required here, a depolarization. That seems to be the key to the question, doesn't it? Because we have depolarizations and hyperpolarizations represented in A, B, C, D, E, some with contraction and some with relaxation. So, a ryanodine receptor agonist is something that would mimic calcium, wouldn't it? Because calcium is released from the ryanodine receptor. It's a calcium channel, and so something that stimulates the ryanodine receptor calcium channel would be an agonist and would mimic then the effect of calcium coming into the sarcomere and causing the cross-bridge cycling for active tension and purposeful movement. So, if we administer the ryanodine receptor agonist, aren't we bypassing the nerve depolarization that results in release of acetylcholine, attachment to acetylcholine receptors, and depolarization at the Z lines of the T tubules that's going to release the calcium. So it seems like we would get contraction because calcium is forming the cross bridges, but it seems like it would be without depolarization. So that jumps out at me as being the best answer. But remember, in a multiple choice question, we can't always stop there, because sometimes there are two choices that are correct, and we're always required to select the very best one. So let's see if we can eliminate the others using our logic. Contraction with depolarization, I think clearly we can eliminate, because we said that when we administer the ryanodine receptor agonist, we're bypassing the depolarization that comes from the nerve and the muscle, what we call excitation, contraction, coupling. We're eliminating the excitation phase of the excitation, contraction, coupling in this experiment. So it seems like D is not appropriate. B, contraction with hyperpolarization. Well, hyperpolarization of cells that are excitable inhibits the excitability. In other words, makes them less excitable. And so when we're talking about contraction with depolarization, we're talking about increasing the excitability of the muscle for contraction, and it seems like we can eliminate hyperpolarization, which would inhibit excitation and cause inhibition. So A and B seem to be out. Now D and E are both dealing with relaxation. If you know anything about that ryanodine receptor, if we release the calcium, we're going to get contraction, not relaxation. So D and E are eliminated, and we're stuck with C as being the most appropriate and very best answer.